Hi, my name is Malay Haldar and I'm an assistant professor and a physician scientist at the University of Pennsylvania and a Lloyd J All-Star. We have several interesting observations, some of which is published and some of which we are still pursuing in terms of different microenvironmental factors in cancer that affect differentiation and development of myeloid cells. So one of the published work relates to retinoic acid, and what we found is that many tumors produce high levels of retinoic acid, which then acts on a type of myeloid cells called monocytes and has a profound impact on their differentiation into either one of two key immune cells, dendritic cells or macrophages. What we discovered is that tumor-derived retinoic acid actually promotes monocyte differentiation into macrophages, and these macrophages end up helping the tumor cells proliferate and metastasize and escape immune responses. And at the same time, the retinoic acid prevents monocyte differentiation into a dendritic cell, and that actually leads to immune evasion, because dendritic cells are important for initiating anti-tumor immune responses. So by extension, what we are right now pursuing is to develop ways in which we can block either retinoic acid production from tumors or sensing of retinoic acid by the myeloid cells. And we envision that either one of these approaches, or perhaps in combination, they would strongly alter monocyte differentiation in a manner that it ends up helping anti-tumor immune responses. And we think this has a strong potential to synergize with other more common types of immunotherapies that are currently being used. So that's one recent discovery, and we have other interesting discoveries around different types of dendritic cells that are present in the tumor microenvironment. And a recent finding in the lab has been the role of interferons in generating these unique dendritic cell subsets. And we also have findings relating to macrophage diversity and interesting subsets or subtypes of macrophages that either help tumor growth or prevent tumor growth, and we are pursuing these findings. So in two ways. So one application of our discoveries is completely novel types of immunotherapy by targeting a cell or cells that are currently not being targeted by immunotherapy. So that's completely novel. And the other application is to actually, like I said before, synergize with existing forms of immunotherapy. As an example, there are most of the current immunotherapies actually target T cells, and T cells are very good at recognizing cancer and destroying them, or recognizing pathogens and destroying them. However, they work in lockstep with another important immune cell type, which is macrophages and dendritic cells, or mononuclear phagocytes. So we can help these T cells and also prevent them from getting shut down in the tumor microenvironment by targeting these myeloid compartments. So we are thinking ahead. We are considering both novel forms of immunotherapy as well as to support and bolster current types of immunotherapy in cancer. So as I briefly alluded before, we are very interested in developing new inhibitors that specifically target the retinoic acid pathway. So that's something that is currently underway. And we are also exploring the types of tumors that would be vulnerable to these retinoic acid based therapeutics. One thing we have recognized since we published our paper is that not all cancer cells depend on retinoic acid for immune evasion. There are some tumors that do that, and then there are many others which don't. So we need to identify tumors that are vulnerable to these inhibitors. And also we are looking for specific aspects of the RA pathway that are being used by these tumors. What I mean is, if a tumor is dependent on high levels of retinoic acid production for immune evasion, there are many enzymes that are important in that retinoic acid synthesis pathway, and different tumors use different enzymes. So we're trying to figure out what enzymes are being used by which tumor types, so that we can then properly target those tumors. So that's one angle we are taking. The other translational approaches that we are, or translational projects in the lab, relates to targeting dendritic cells and macrophages. As an example, we recently found that there is a subset of macrophage in the tumors that are preferentially localized very close to blood vessels. And these macrophages are involved in new blood vessel development, which is very critical for a growing tumor, because a growing tumor needs nutrients, and so they depend on vasculature for that. So these macrophages help tumors grow new blood vessels. We have also found that these macrophages are immunosuppressive in a certain way. The important thing is we found that these macrophages express a receptor called endothelin receptor B on their surface, and endothelin signaling appears to play an important role in the functions of these macrophages in terms of helping angiogenesis or immunosuppression. So there are inhibitors of endothelin receptor B that can be potentially used for targeting these macrophages, either alone or in combination of immunotherapy. So that's another angle that we are taking, and then there are several other projects in the lab that relates to that takes a similar approach, which is to find a novel subset or an interesting subset of these cells in tumors, find out what they're expressing, and then within that identify things that can be therapeutically targeted and then go after it. So that's been the overall approach in the lab.